Our final speaker is Stefano Rensi. Stefano is a research engineer at Stanford, and the title of his talk will be Repurposing Existing Drugs to Fight COVID-19.
Hi, Russ. Thank you for the introduction. Okay, so COVID-19 is a global pandemic that's already killed over 40,000 people, and it threatens to overwhelm healthcare systems around the world. Now, we desperately need treatments for it, but we don't have the 5 or 10 years that it normally takes to develop a new drug and get it into clinical trials. Now, the fastest way to develop drugs is to repurpose existing drugs that are already on the market or in clinical trials. The big advantage of this approach is that you can leverage a lot of work that's already been done. These compounds have already undergone extensive testing, and we know a lot about their safety. So if we have good reason to believe that they're effective, we can advance them into clinical trials almost immediately. Now, today, I'm going to talk about some work we've done using natural language processing, protein structure prediction, and biophysics to identify drugs that can be repurposed to fight COVID-19. So the first thing that we did was mine the literature for therapeutic targets. The goal here was to develop a working hypothesis. What protein are we going to target? Why do we think it would be effective? And what evidence do we have that this therapeutic strategy would actually work? We did this using a tool that we developed in our lab called Docs2Graph, which uses natural language processing to generate a map of relationships between chemicals, diseases, and genes discussed in a corpus of documents. Here, the document corpus that we used are the abstracts from the top 15 articles returned by a PubMed search for SARS and all the articles that they've cited. The genes are green, chemicals are yellow, diseases are red, and the size of each dot represents its importance in the corpus. Now, the thing about the maps generated by our tool is that they also work as a content indexing system that lets you drill down on documents and even individual sentences of interest. So when we click on this gene TMPRSS2, we get sentences that tell us that it is a host protease expressed on the surface of cells that line the airway. It activates a mild human coronavirus, 229E, by cleaving the spike protein. It's been shown to facilitate the spread of SARS and Middle East respiratory syndrome coronaviruses in the airways of mice. So now we have a promising target and a mechanism of action, right? This gene or protein, TMPRSS2, cleaves and activates the coronavirus spike protein, which allows it to bind its receptor, ACE2, and enter the cell. We want to disrupt this process by inhibiting the activation step. To do this, we need to find existing drugs that can bind and block the active site of our target, TMPRSS2. We decided that the best way to do this, given the available resources, was to use a biophysical model to model the binding interactions between our target protein and potential inhibitors. So here, what I'm showing you is an ensemble of homology models that we generated for our target. You see, the actual structure of the protein is currently unknown, so we had to use computational models to make predictions about it. And to do so, we used the known structures of closely related proteins as templates. So you can see here that all of the models align pretty well with a little bit of variability in the loop regions around the active site. Now here, I've highlighted the active site of the protein. This is the part that catalyzes the activation of the coronavirus spike protein. Now we want to block this with a small molecule inhibitor. And here you can see a close-up of a small molecule inhibitor that is happily bound in the active site of TMPRSS2. And finally here, if you look at the dotted lines, these show some of the physical interactions between the active site and the inhibitor that keep it bound in place. So these are the interactions that we're going to be modeling in our biophysics simulations. Now, the type of simulations we're going to use are called molecular docking. And basically the way this works is that you take a potential inhibitor and you jam it into the active site, and then you look at how well it fits, as well as the number and strength of physical interactions. Doing this, you can use some principles of physics to calculate the binding energy, which indicates how strongly that potential inhibitor is going to bind the active site. So we did this for 18 molecules in total. 3 of them were designer chemicals that are known to be very potent inhibitors of TMPRSS2, and we used these as positive controls. The other 15 were our most promising repurposing candidates. So this slide here shows the results of our docking simulations. The horizontal x-axis shows the binding energy. Negative scores, so Things on the left side of this graph are good. They indicate tight binding. Each entry on the vertical y-axis is a compound, and the box and whisker plot associated with each compound shows the distribution of binding energy scores from docking simulations. So here we've highlighted the 3 positive controls in red, and they all rank very highly, which is good because this is what we would hope to see if our simulations are working properly. We also see that some of our repurposing candidates, which I've highlighted here in green, have binding energy scores approaching the positive controls, which suggests that they can also be potent inhibitors of TMPRSS2. Now, out of these top 6 candidates, 3 of them are currently marketed, meaning that they've been approved for use in humans and they're being prescribed to patients for other things. The other 3 are not approved, but they've made it pretty far into clinical trials. So while they're not available for prescribing, they've already been cleared for testing in humans. Now, as it turns out, this candidate here that I've highlighted in red that we predicted to be a potent inhibitor was reported to inhibit entry of the Middle East respiratory syndrome coronavirus into human lung cells, and it's currently being tested in clinical trials in Japan. So that's pretty exciting for us. One thing I would like to make very clear is that these are preliminary results. When you look at the hierarchy of evidence, computational predictions are pretty close to the bottom. You can use them to figure out what experiments you should do in cells and mice, but it would be very irresponsible to test these compounds in humans without further experimental validation. That being said, we're very excited about these results, and we're currently testing our candidates in cell models. I would like to acknowledge the organizations that fund my work, as well as my co-authors, current and former lab mates, and a long list of researchers at other institutions who I've had the pleasure of working with. Thank you very much. And if there's any time left, I would be happy to take a question or two from the moderator.